Biotech Park, Lucknow, and Chairman of NASI Lucknow chapter to please deliver the formal welcome address.
धन्यवाद सुचित्रा जी. Professor G. Padmanabhan, President of the National Academy of Sciences, India. Professor Manju Sharma, Chairperson, NASI New Initiatives. Professor Paramjit Khurana, General Secretary, Outer Station, NASI. Professor Veena Tandon, Professor Madhu Dikshit, Professor A.K. Tripathi, Dr. Rituraj, Dr. Suchitra Banerjee. I see Professor Kamboj and Professor Ashish Datta, the past presidents of NASI, distinguished fellows and members of NASI, Dr. Neeraj Kumar, Ms. Archana Pant, dear participants, ladies and gentlemen. It gives me immense pleasure to welcome all of you to this very important webinar on bioresources for combating COVID-19. We highly appreciate and feel privileged to have in our midst Professor G. Padmanabhan, the President of NASI and former Director of Indian Institute of Sciences, Bengaluru, and Professor Manju Sharma, NASI DST Distinguished Women Scientist Chair Professor, past President NASI, and former Secretary of Government of India. The nation owes to both of them for many new initiatives, leadership, and policy formulations. We extend a very warm welcome to you, Professor Padmanabhan and Madam Sharma. We are truly delighted that we have distinguished speakers this morning. Welcome to this webinar. 10 months ago, since we first heard of COVID-19, the pandemic has claimed more than 1 million lives and infected more than 38 million people in 190 countries. Infections are rising and there are troubling signs of new waves. Much about the virus remains unknown. One basic fact is clear. The world was not prepared. In India, 83.7% of confirmed cases of COVID-19 patients have been discharged, 11.12% are active, and mortality rate is 1.52. Timely action by Government of India has reduced the impact of the Indian scientists, medical professionals, and industries are working on war footing for developing fast, cheaper, and reliable methods for vaccine development and cure of this dreadful disease. Towards this goal, the present seminar aims at discussing useful bioresources having a role in combating COVID-19. Let me also take the opportunity to share that NASI Lucknow chapter is regularly organizing awareness programs, lectures by eminent experts for creating awareness about the pandemic. Dr. Praveen Tandon, the masked man of now, delivered a lecture on I Protect You, You Protect Me for school children that evoked very lively interaction. Professor Shailen Saxena, Professor and Head, Center for Advanced Research, King George's Medical University, delivered a lecture on pandemic novel coronavirus disease, the unseen and unsaid for biology students and researchers. Professor Suryakant, Head Department of Respiratory Medicine, King George's Medical University, spoke on COVID-19. Immunity is the best vaccine for researchers and general public. Our Prime Minister has given a nationwide call to observe September as POSHAN Abhiyan month. Dr. Manju Sharma has advised all the investigators of NASI projects for tribal populations to take up a special program on nutrition, focusing on children to combat malnutrition, give them more immunity towards COVID-19, and help them to do better in their studies. Following this call, we recently organized a lecture on Sishu Poshan by a famous pediatrician of Lucknow, Dr. Shalini Tandon, for the tribal people of Eastern UP. In order to bring nutrition, it is posted to the center stage of the societal programs of NASI, the present webinar is one in the series of the programs to be organized across all the chapters of NASI. Once again, a very warm welcome to each one of you and look forward to meaningful deliberations. Thank you so much for joining us. Now it's over to Dr. Suchitra Banerjee.
Thank you, Professor Tandon. for very nicely elaborating the different activities that's going on through NASI Lucknow chapter. Now, may I request Ms. Archana Pant, young woman scientist NASI, to brief about the NASI's POSHAN programs that is coming under this webinar, please.
Thank you, madam. A very good morning to all of you. On behalf of NASI headquarters, I welcome you all to this webinar, which is a joint venture of NASI headquarters and NASI Lucknow chapter. To begin with, we must express our gratitude to Hon'ble Madam-Dr. Manju Sharma'ji for all her efforts to initiate this nationwide program on a very crucial aspect that deeply concerns our lives as well as a very important topic of national development. And that too during a time when the entire humanity is in the grip of the global pandemic, COVID-19, and especially the people in a country like India need to be made aware and also educated on this very important aspect, that is nutrition, which plays a very important role in enhancing our immunity, especially in the fight of the pandemic. We are also very much thankful to our Honorable President, sir, President of NASI, for his very kind approval and also motivation to implement this program all across the country. As some of the students have also joined us on this occasion. So before coming to POSHAN program, I would just like to mention in a very nutshell that the National Academy of Sciences India, NASI, is the oldest science academy of this country, founded by Professor Meghnad Saha, who was an eminent scientist, an academician, and above all, a great social reformer, who used to believe that the problems in a country like India could only be solved with the intervention of science and technology. And that's why he thought of establishing a forum for all the scientists so that they could discuss their problems and provide scientific solutions to those problems. And this is how NASI came into existence in 1930 with its mandate Science and Society. And since then, NASI has been implementing several programs on science popularization, awareness, also some programs on capacity building and skill development, and also addressing some subjects relevant to national importance. We have different programs meant for specific groups of the society. We have programs on technological empowerment of women. That program is meant for women scientists, researchers. We have programs for rural women. We have programs for students, the research scholars, on how to write a scientific paper. Then we have programs for startups or entrepreneurs. We also have programs for science teachers. We have programs for tribal people and also for the defense personnel. So this is how NASI is trying to reach every state of society and trying to address the problem of society. Not only that, the fellows and members are providing very much support, and also the local chapters for providing a scientific solutions to those problems. Now coming to the POSHAN program, I would like to just brief that NASI has been already addressing the problem of malnutrition since several years. So this program, the problem is not a new for us, and still continuing to address this problem. We have also executed a project in joint collaboration with ICMR. The project was executed at 3 places: Hyderabad, Allahabad, and Vadodara. And we also got crucial recommendations for implementation. Now, after this, the announcement of our Honorable Prime Minister, as Chandran just mentioned, Honorable Madam gave us a valuable suggestion to initiate this program on nutrition, that is on POSHAN. So NASI came up with this idea to start this program all across the country under the chairmanship of respected Madam. And as a first step, we started a series of webinars to enhance the spread awareness and also educate, especially for the students, so that they could know more about this nutrition and cope up with the problems during the pandemic. We have also submitted a proposal on nutrition to the Department of Biotechnology. The project aims to develop some edible food products for the especially for the underprivileged section of the society, and also they are being educated and they are being provided imparted training to develop the project and also awareness on nutrition. So with this effort, NASI looks forward to spread more awareness all across the country and we look forward to have more such events so that especially the students and women must know about nutrition and able to cope up the problems of malnutrition. So with these few words, I hope that this webinar will also be very much beneficial for the students and they will be listening to the learned speakers and the speakers will enlighten them. And with these few words, now I request Dr. Banerjee Madam, the coordinator of this program, to please throw some light on the role of the local chapters, especially the Lucknow chapter. Madam is NASI Senior Scientist from CMAP and also the Secretary of Lucknow chapter. Thank you. Over to you, ma'am.
Thank you very much. Thank you very much, Archana ji. It's a great privilege and honor for us, the Lucknow chapter, to get this opportunity to organize this seminar under this chapter. As Archana ji has already narrated the different mandates under the POSHAN and the other mandates of NASI headquarters, and Professor Pramod Tandon has very nicely elaborated that what are the different activities that NASI is taken up, NASI Lucknow chapter has taken up for dealing with this pandemic. I will come directly to the concept of this present webinar. Actually, as we all know, the intents of all the local chapters are like the functioning of the finer arteries carrying oxygenated blood from the heart to the distant part of the body. So, with this concept, Professor Manju Sharma'ji has visualized that Lucknow chapter would be the ideal to carry on this webinar because of certain reasons. We are indeed very, very grateful to you, Madam, for giving us this opportunity. And we are also indebted to Professor Padmanabh for allowing us to hold this meeting through Lucknow chapter. Now, as we all are aware that this unprecedented to each and every individual, focus attention on own nutrition for immunity boosting. And we all know that people are strongly adopting daily use of nutraceuticals. I emphasize nutraceuticals. As new part for 2020, use of time Crickets, Guduchi, Ashwagandha, Ayush 64, so on and so forth. We all know that there is probably not a single house left which is not using the very widely accepted kada, which is changing with the changing ingredients of tulsi, haldi, giloy, etc., etc. But people are being misguided as well, not being able to understand how much and what extent they can use this. As a result, there is reports in the news media as well that some adverse reaction side effects are occurring. And there are cases where the jaundice-like situations have arrived with normal liver profile. So it is very important to realize that knowledge-based awareness for the best utilization of the first medicinal plant repository is the need of the hour. And for that reason, probably, Professor Manju Sharma'ji has visualized that this would be the right chapter to convey this message. To fulfill the national need, we have found this bioresource for combating COVID-19 is the ideal way to convey many messages. As we all know, that it is very important to highlight that as the COVID-19 is a new entity, many things are unknown, as Professor Tandon has already said. It's very difficult to understand in terms of its drug receptors and appropriate drug designing. as well as repurposing of many age-old phytomolecules have started widening their therapeutic applications, as in the case of current entries of artemisinin, reserpine, withaferin A, etc. in anti-cancer molecules beside their conventional uses. Some new therapeutic avenues are expected to open up for combating COVID-19 through actual research in the area of medicinal plant resources. So today's webinar can serve the prelude of the mega endeavor of Honorable Madam Manju Sharma'ji under NASI's new initiative in opening up a Pandora box of therapeutic solution for tackling this global pandemic. The Lucknow chapter is very fortunate to get this opportunity, as I'm telling repeatedly. Lucknow is known as a city of institute and is proud of owning 4 CSR institutes, among which 3 are majorly involved in medicinal plant research, and the 4th one is also indirectly complementing them. All the speakers are representative of the national laboratories of repute in medicinal plants. So what can be better than to learn it from those eminent scholars and eminent scientists of this medicinal plant theme. Incidentally, this opportunity is also bestowing upon us the privilege to fulfill another directive of our Prime Minister, that is being vocal for the local. Finally, I would like humbly to acknowledge the NASI Lucknow chapter's gratefulness to the President Padmanabhan Sir, respected Madam Manju Sharma'ji, distinguished members of NASI Council for giving us the chance to be associated with NASI headquarters in organizing this webinar. My warmest wishes to all of you and my pranam to my seniors for the forthcoming Navaratri. Thank you very much. Now, after concluding this one, I have a great privilege and honor to request Madam Manju Sharma'ji to bless this webinar with her opening remark. I know that she doesn't need any introduction in all that fraternity, scientific fraternity, but for the benefit of the students who are probably new in this area, I need to illustrate some of the glimpses, which is very difficult to sum up in few words. Professor Manju Sharma'ji is the dynamic biotechnologist who has played a significant role in shaping biotechnology research and application in the country. She is the past president of the National Academy of Science India and also Indian Science Congress Association. She was the Secretary DBT, Ministry of Science and Technology, and she is the one who brought the biotechnology to this condition where India is leading today. She is the NASI DST Women Scientist Chair Professor and Chairperson of NASI New Initiatives. She is well recognized for her many-fold significant contribution in the area of biotechnology, as well as for the welfare of women scientists and women particularly, including tribal populations who regard her as the role model. She promoted the Biotech Consortium India as a novel institute for science-industry interface. She is the unique contributions in developing several institutes under DBT. She is the recipient of a large number of awards and honors, to name a few, starting from the Basbik Award, Norman Borlaug Award, Om Prakash Vasin Award, to the very recent ones, like the Lifetime Achievement Award of Human Service in Biotechnology for the year 2018 from Riva University Bank, and the Lifetime Achievement Award 2019 by Society of Entropharmacology India. She is the recipient of the Lifetime Achievement Award of the Biospectrum and was awarded Padma Bhushan by Government of India in 2007. It is indeed an honor and privilege, Madam, for me to invite you for blessing this occasion through your opening remarks, which is your brainchild, this whole webinar. Thank you very much. To Madam Manju Sharma'ji, please.
Madam needs to unmute. Madam, please unmute yourself.
Is it okay?
Yes. Yes, it's okay now. Yeah.
It's okay now. Okay. Honorable Professor Padmanabhan, the President of our Academy, National Academy of Sciences, Professor Pramod Tandon, the chairman of the local chapter, Lucknow, other very distinguished scientists who are all connected in some way or the other to NASI, and they are past presidents, very distinguished fellows, and Dr. Paramjit Khurana and Dr. Madhu Dikshit and Professor Veena Tandon. I'm taking the names of all the distinguished women scientists also. Okay. I don't have to talk too much about either COVID or the nutrition issue which Professor Pramod Tandon and Dr. Suchitra Banerjee have highlighted. But I want to focus my brief comments on the bioresources of this country. India is bestowed with enormous biodiversity and bioresources. We have a hotspot of biodiversity in our country, two hotspots rather, the Northeast and the Western Ghat region. More than 55,000 plant species, 33% are endemic, and this gives us very good opportunity to work on the bioresources and see how well we can divert many of our bioresources and convert them through biofortification, through SNT intervention for nutritious food for the poor people. It is, I'm not going to elaborate the position India has. All of you know, and especially our president, he is very, very concerned about the malnutrition, the largest number of women and children in the world, and our hungry children who are in the rural areas, who are unable to get even education properly because their body is not healthy, therefore the minds are not healthy, and accordingly they are very mediocre and poor in education. We have a very large population in rural areas, and we have to pay more attention. And NASI, as per the mandate given to it by its founder, Dr. Meghnad Saha, Science and Society, NASI is trying its best. I can say that we can't do everything, but whatever small effort we can make, I think we are trying our best to contribute towards creating awareness. The word Jagrukta, which Hon'ble Prime Minister has used, creating awareness, and also observing September 2020 as a POSHAN Maah, because the Prime Minister feels that we have to send this message across the country, both the malnutrition condition and to educate particularly for the education of the children, they have to be very healthy. And secondly, awareness about the COVID-19 pandemic. We have been thinking, and in various seminars, some very good ideas have come up, and nutrition garden is one of the very important suggestions which emerged in one of our webinars. Now, we have been also promoting horticulture crops, which are very nutritious, should be grown by the farmers in addition to the agriculture crops. We have the agri-economy in the country, and we want this agri-economy to move towards bioeconomy. I think all of us understand this bioeconomy. We have talked about it on several occasions, and more than 60% of our rural population is still depending on the farming practices. We have to introduce these new concepts that they should have a small horticulture garden, or they should have a nutri garden, things like that. And what are the nutritious crops, whether it is palak, or it is carrots, or it is green peas, or it is even brinjal, all of these, including potato, they have the nutritious value, and either protein content is very good, or carbohydrates they provide. I mean, they provide everything which is required for a balanced diet, which our children require. between during this year 2020, we have produced more than 290 to 295 million tons of food grains. Now, the question is it possible that through our meager efforts, and of course, the other scientific institutions, ministries, they are all working on this, and certainly the Ministry of Women and Child Development, can we convert this very large food grains which we have produced to nutritious foods and create awareness amongst the farmers, amongst the villagers, that what is the nutritious food they should have. In one of the papers I was reading that ragi is one of the most nutritious crops. So many of the millets, bajra, jowar, all these are very important. So we have to start educating and creating awareness amongst the farming community that they should eat this. You know, there is a saying, and I'm sure all the people in UP would understand. Sorry, Professor Padmanabhan, it's in Hindi, but there is a very famous saying that sab kuch hai sansar mein, sab kuch hai sansar mein. Means everything is available in this world. Karamheen nar pavat nahi. जिसका भाग्य ही नहीं है उसको मिलता ही नहीं है. अब ये भाग्य जो है ये हमें अपने बच्चों का, विलेजर्स का, विमेन जो प्रेग्नेंट विमेन हैं, गरीब औरतें हैं, उनके लिए इस भाग्य वर्ड को चेंज करना है. उन्हें हमें कर्महीन नहीं बनने देना है. तो जब हमारे पास इतना फूड ग्रेन प्रोडक्शन है तो हम लोग क्यों नहीं बायोफोर्टिफिकेशन और इस तरह के मेथड्स यूज कर सकते हैं. आई वाज वेरी पर्टिकुलर दैट डॉक्टर Anil Tripathi and Dr. Pramod Trivedi from CMAP, all these people attend because they have so much knowledge of the bioresources of this country. And they guide several groups in India. And I have discussed this matter with the Department of Biotechnology, even with ICMR, and they are willing to support projects provided we come forward and do something. So my request to all the very senior and distinguished people here is let us take up some more. NASI can do very small effort. NASI cannot do everything because we don't have too many people. We are only dependent on our fellows. But the other distinguished scientists, they can take up programs and go send teams to the villagers to think that they can have a horticulture garden or a nutrition garden, and how do we go about using the bioresources, the plant resources for the welfare of the poor people. The last thing I want to say here is that we are also very keen for an industrial development. Prime Minister has given several initiatives like the startups, Make in India, a skill improvement and development, all these initiatives. So India wants to go with the industrial development. But when I look at the whole situation, this is my personal view, I feel as Father of the Nation, Mahatma Gandhi had said, that we need to develop, we need a bio-industrial development rather than just an industrial development. I repeat, we need a bio-industrial development rather than just an industrial development. Because if we want to set up industries, they have to be dependent on the bioresources, especially in the village areas, the small-scale industries, medium-scale industries, MSME, all these. You know, we can do very advanced science also with using our bioresources. So I'm not going to say more about this. What request to all the very distinguished people here, and London has already initiated very good programs from the Biotech Park. Professor Anil Tripathi can take up, Professor Trivedi in CMAP, and Dr. Alok Dhawan, and many other, all of you in fact, Dr. Rai, we should start a large number of projects. And what I want is NASI fellows, NASI officers, they take the lead. Finally, my request is that we use science and technology interventions through bioresources for developing bioeconomy in this country. It is just not a slogan, bioeconomy. We can move and we can go ahead with this. And that's for the population, welfare of the pregnant women, the very poor women, anemic women, and the children. So that's what I want to say. And I'm very glad that chapter has done this work, and my best wishes for the success of this webinar. Thank you.
Thank you very much, Profuse. Thank you from the bottom of our heart, Madam. It's literally a guidance and light for us to how to go forward through your all the suggestions, like showing us the S&T intervention, bio-industrial developments for developing bioeconomy, and how to change the fate of the children through this, taking it forward through different scientific endeavors. Now, it is another huge task for me to introduce the next speaker for his very kind blessing on this occasion, that is none other than the President of NASI, Professor Padmanabhanji. It's literally, I don't know where to start and where to end. Professor Padmanabhan is literally an instrumental in ushering the recombinant DNA technology in the country. He studied the heme biosynthetic pathway in different eukaryotic systems and discovered the heme biosynthetic pathway in the malarial parasite and showed how it can be a unique drug target. He showed also that the curcumin from turmeric has anti-malarial activity and embarked on clinical trials to prove the efficacy of curcumin-based combination therapies for malaria. He worked closely with several government agencies to further the cause of biotechnology in the country, as I have discussed with relation to Madam Manju Sharma'ji as well. He helped and promoted vaccine industries in the country and supported his colleagues in the development of recombinant hepatitis B vaccine and DNA rabies vaccines. He chaired several committees of DBT and was part of several initiatives to promote life science research. He was a member of the WHO group on malaria for 3 years and served on the INSA Council. Professor Padmanabhan was recipient of Padma Shri and Padma Bhushan from Government of India, Shanti Swarup Bhatnagar Prize for Science and Technology, and Jawaharlal Nehru Bharat Centenary Award from INSA. He is also recipient of the C.V. Raman Research Professor of INSA. He was elected Fellow of the Indian Academy of Science, Bangalore, National Academy of Science, and for the Science for the Developing World. And for his awards and other things, I cannot say anything over here because it's a huge list. It's literally very difficult, sir, to brief your endeavor in science over here in these few words. May I now request you for blessing this occasion through your inaugural address, please. Professor Padmanabhan, sir.
All other eminent speakers and the participants are requested to kindly mute their mic. Thank you.
Thank you, Suchitra, Dr. Manju Sharma, Dr. Pramod Tandon, and all the distinguished speakers of today, Dr. Veena Tandon, Dr. Madhu Dixit, Dr. Who else is speaking today? I should not take names because definitely I will be omitting some names. Dr. Paramjit Khurana's secretary, and very many distinguished directors of CSIR institutes, past, present, presidents and past presidents of NASI. I cannot, but any number of times we can thank Dr. Manju Sharma for her vision, for really implementing the vision of the founder, to take science to society. She always comes up with newer ideas, vision, and this is something which the Prime Minister himself, POSHAN, I remember. And this one, I should congratulate the Lucknow chapter for coming forward, Dr. Pramod Tandon, Dr. Suchitra Banerjee, for this wonderful meeting, and getting wonderful speakers like Dr. Anil Tripathi and others I have mentioned. Well, a few thoughts I have. Dr. Manjuji has already talked about bioindustry, bioeconomy. I thought I'll just briefly talk about COVID. There is no vaccine. There is no drug available. I chair the committee for vaccine development, all the things which have been supported in the country. Sometimes I feel there is an over-expectation that the vaccine is going to solve the problem. Of course, vaccines are preventive, but let me, from all the experience we have, we know smallpox is a rare example. It's not, polio is another example. But a disease like COVID cannot be eradicated by vaccine. It will kind of, prevent or to some extent. And there is no drug available, as we said, which is an after event, basically. So what is it that will really be of importance is nutrition. It's very clear, it doesn't need any rocket science to understand that nutrition is the one that will actually prevent even an enormous pandemic like COVID. Why? You can clearly see literature. For example, you take obesity. You may be surprised generally. Why is it that advanced countries like Europe and US are so much affected by COVID? And they have been affected earlier also by the earlier viral disorders. Why? And their hygiene is much, much better than India. We are a poor country. Why is it? To my mind, it may not be absolutely right, but I do feel it's very important. If I go through literature, I find nutrition, very rich nutrition, high in salt, high in fatty acids. They link obesity, for example, to many of these saturated fatty acids. And what is fundamental to all this is inflammation. Inflammation, is whether it is infectious disease, whether it is cardiovascular, whether it is any kind of disease. Inflammation actually is a protective response. But what really, as Suchitra said, I work on malaria, is one example only. There is inflammation, but it's uncontrolled inflammation. And that is what you call, it leads to cytokine storm, as we call it. If you don't prevent inflammation, you are getting into ARDS, acute respiratory syndrome, in COVID, and people die. So one of the approaches has been how to prevent this cytokine storm, how to prevent this inflammation. And I think, the best way to prevent inflammation is to have a proper nutrition, appropriate nutrition. Now, this is very, very clear why vegetarian diet, or, I can't propagate, I am a teetotaler, but, but generally you see why vegetables, in Western world compares, not the India, they compare with Mediterranean diet. Mediterranean diet versus US diet, for example. Mediterranean diet, which is very rich in vegetables, very rich in fruits and so on. So if you really look at what are the elements which actually do, this is polyphenols, for example. There are many, for example, Suchitra again mentioned curcumin. One of my interests have been curcumin. Phenomenally anti-inflammatory. phenomenally anti-inflammatory. We have so much of data published, etc. But unfortunately, many like curcumin, many of the plant products are poorly bioavailable. That has been an issue. I think many of us should concentrate how to improve the bioavailability of many of these natural molecules. If you really look, there are very few, like artemisinin, which can directly go and kill. But most of our molecules, including curcumin, build the host defense. Now, host immunity, as we know, this is where there are many, whether it is TLR4 based innate immunity, whether preventing IL-6, which is the cytokine, which is the inflammatory cytokine. You can clearly see all these molecules are capable of doing that. The only issue is how do we make those bioavailable? I'm not saying at the molecular level. It has to be a combination of many things which should, diet itself should be, because questions are asked, people have been taking turmeric, people have been taking, it's not, anyway, that's a different issue altogether. But the bioavailability is one issue which Western world also tries to put down. If you say, for example, why they put down Ayurvedic drugs and things like that, or they say this is toxic, or they will say this is not bioavailable. They have not understood the issue. They have not understood the problem. And there are today, many technologies are available for improving bioavailability of many of these natural molecules, which are essentially anti-inflammatory. I must tell you again and again, inflammation is fundamental to many of the issues. And that is where, the immunity needs to be built, whether it is innate immunity, whether it is adaptive immunity. There are many parameters through which one can do. And a simple element like zinc, for example, I can tell you, is so powerfully anti-inflammatory. It affects the immune system, whether it is monocytes, whether it is macrophages, whether it is lymphocytes. Zinc nutrition is so important. Perhaps some of you remember our previous secretary of DBT, Dr. Ban, late Dr. Ban. He propagated zinc nutrition of children in a very big way, and I think this is now an All India program. Under the biochemistry, we know zinc is one element is present in so many proteins, and it is very closely related to immunity. It may be known or may not be known. Even amino acids like glutamine play a very important role in immunity. And the last thing I want to give is vitamin D. When people meet like old people like me, they say, "Are you taking vitamin D?" Now, vitamin D, we know vitamin D gets converted into dihydroxyvitamin D3, and there's a receptor for that. And that receptor is present in all the immune cells. That is why vitamin D becomes very important to prevent inflammation. So I can go on, exhibiting whatever little knowledge I have. All that I want to tell you is all the natural products, all the bioresources which Manjuji mentioned, are the ones which can give us a balanced diet. And perhaps nutrition is the best way of preventing many of these. They may not be able to solve the issue, but definitely they can prevent it. And it has to be a balanced diet. Polyunsaturated fatty acids, or there can be many other ways by which they do, and they provide many of these molecules. At molecular level, I can tell you there are hundreds and thousands of molecules, mechanism of which we have not understood in India. And we are very rich in medicinal plants, the CMAP, the directors are there. There is a need, in my opinion, to understand, rather than looking at synthetic drugs, it is important to look at this natural molecules, their mechanism of action, more and more. Not even in isolation, perhaps. After, I must tell you, last point before I conclude, I battled for 10 years with the Drug Controller General of India's office to get approval for clinical trial with curcumin. You will not believe, as a combination drug. 10 years. Because they thought curcumin is a natural molecule, artemisinin is not. Because artemisinin gets converted into artemether. Therefore, it is a molecule. It's a drug, whereas this is a natural extract. Only 3 years ago, I think the DCJ approved another route. We can have plant extracts, provided they have at least 4 molecules and they have characterization criteria, but they are now allowing. So I have now the support of NIMR, National Institute of Malaria Research. They are the ones who have to do the trial, and they are ready with all these trial to do. But unfortunately, the hospitals in which the trial has to be done have become COVID hospitals. So we are waiting for for the signal. Yes, COVID has come down, so you can do other trials like malaria and other disease trials can also be done. Therefore, I think I'm only putting few things in a different way than what Dr. Manju said. What is very, very important is our natural wealth, natural food, and I feel how best to promote them. And if we have scientific data on the molecules which they are there and how they work and how they affect, even let me host immunity build-up. That's the most important criteria to prevent disease, whether it's infectious disease, whether it's cardiovascular, whether it's any other kind of disease. I once again want to congratulate the Lucknow chapter for organizing this particular meeting to combat COVID-19 with bioresources. And the other dimension of bioresources, Dr. Manju has already pointed out. Let me congratulate Dr. Parmod Tant and Dr. Suchitra Banerjee and all the speakers today, and very well for this meeting. Thank you so much.
Thank you very much, sir. I cannot say you thank you. It's our utmost gratitude to giving us the direction of being in the medicinal plant science for the last 40 years. So I am so much indebted, and we all are, to get that future direction, where we should start and where we should go, particularly from a person who has developed recombinant hepatitis B vaccine and DNA rabies vaccine, who is giving the direction for the COVID-19 in terms of the medicinal plants use, their mechanism of action, synergistic combinations of different medicinal plants use, as well as to find out how the new molecules can act on this new virus. It's a unique direction for us, for the whole India, as well as I think it will go far, far ahead globally. We are indebted and we are profoundly thankful to you, sir. Now, coming to the next next point of the program, it's going to be the lecture by the 3 eminent scientists. And for that, I would like to invite Professor Beena Tandon Madam, who is the NASI Senior Scientist Platinum Jubilee Fellow and formerly Professor of Zoology, Northeastern Hilly University, Shillong, to preside over this 3 lecture series. And to give just a brief introduction to her, I would like to say that she has got 41 years of postgraduate teaching and 43 years of research experience, 183 research publications. She is fellow of the National Academy of Science, Indian Society of Parasitology, Zoological Society of India, Helminthological Society of India, and Linnean Society of India. She has received also a number of awards, amongst them, name a few: the Janki Amal Award in Animal Taxonomy, Professor Archana Sharma Memorial Award 2012, Padma Shri 2016 by Government of India, Professor V.P. Sharma Memorial Lecture Award 2019, Outstanding Scientist Award conferred by Chhatrapati Shahuji Maharaj University, Kanpur, and Punjab University Alumni Association Achievers Award in 2019. With this brief introduction, may I now request Professor Beena Tandon Madam to please preside over the lecture series. Thank you very much.
Thank you very much, Dr. Suchitra Banerjee. To begin with, my greetings to all participants of this unique webinar, and special thanks to NASI headquarters and local chapter for having arranged this long-awaited meeting, which gave us an opportunity to virtually meet and see all members of the NASI family. in this lecture series, the lecture session organized for the webinar, as you all know, we have three very eminent speakers who would be interacting with us for the next 1.5 to 2 hours. And I would begin with introducing the first speaker for the day, that is Dr. Madhu Dixit. Dr. Madhu Dixit, who retired as director of CSIR, Central Drug Research Institute, Lucknow, is presently professor in charge biosciences and bioengineering and THSTI national chair. With an illustrious academic career from Kanpur and Allahabad University, Dr. Dixit has been bestowed with many honors and awards. The list is long. I would just name a few. Vaspik Award 2012, Professor Darshan Ranganathan Memorial Lecture of INSA, Vigyan Ratna of Council of Science and Technology, UP, Shakuntala Amirchand Prize of Indian Council of Medical Research, and as early as 1989, she was given Young Scientist Award, Medical Sciences of INSA. She has fellowships of INSA, Indian Academy of Sciences, Fellow of National Academy of Medical Sciences, of course, NASI Fellow too, J.C. Bose National Fellow, member of National Academy of Medical Sciences, and in INSA, she availed visiting fellowship of DBT and INSA costed fellowship to attend the International Congress of Pharmacology, Amsterdam. Dr. Dixit has more than 170 research papers, umpteen number of review articles, has 9 patents to her credit, and a large number of students were awarded MD, MDS, and PhD thesis under her supervision. This afternoon, Dr. Madhu Dixit would be speaking on ashwagandha and human health and COVID-19. May I call upon Dr. Dixit to please start her discourse.
So thank you so much, Veena ji, for your very elaborate introduction. Good afternoon, everyone. Respected Professor Padmanabhan, respected Dr. Manju Sharma'ji, speakers of today's webinar, Dr. Tripathi and Ritu, distinguished NASI fellows, ladies and gentlemen, it is my proud privilege to speak and participate in this webinar. So today I shall be talking about ashwagandha, human health, and COVID-19. I hope my presentation is visible to everyone now. Kindly confirm.
Yes, madam.
Yes, it's okay.
So as I told you that today I will be speaking ashwagandha, Indian ginseng, which is also known as Withania somnifera. So I would today be touching upon the existing knowledge on ashwagandha, which has shown to be anti-cancer, immunostimulant, and also is a anxiety remover. Due to ongoing pandemic and paucity of effective drugs, as was mentioned very rightly by Professor Padmanabhan, various classes of existing drugs are actually being repositioned for and repurposed for the treatment of COVID-19. So Ayush has also initiated clinical trials by joining hands with CSIR and have initiated a clinical trial also on Bethania. Early history of medicine actually dates back from the early days of human civilization. And folk medicines have been derived from plant products and also supplemented by some animal materials and minerals. Not only that, they were also associated with various religious and spiritual healings during the ancient times. And these drugs were discovered through a combination of trial and error and observation of human and animal reactions. Like traditional Chinese medicine system, our Indian Ayurvedic system, Greek medicine, and Egyptian medicines are thousands of years old, and they have been used for various ailments like in infection as well as in other disorders. Why do we prefer natural products? Because they are the best source of drugs and drug leads. Almost more than 60% of the existing drugs either are natural products or there are like suggestions which have been taken from the natural products. So they possess very diverse structures, and they also have been responsible, or they have promoted the synthesis, and their metabolites, secondary metabolites from these sources have been used. So new natural products actually stimulate advances in medicines. And their renewed interest in natural product research is due to the failure of alternative drug discovery in key therapeutic areas such as immunosuppression, anti-infectives, and metabolic diseases. Now, Withania somnifera, which I already told you, is also known as Indian ginseng or winter cherry. It is a terrestrial plant and which has been almost extensively used in the traditional medicinal systems for almost past 30 years. It is not only cultivated in India, but it is also used and cultivated in many other countries world over. Its seeds are usually sown during June and July, and the requirement of seed is around 4 to 5 kg per hectare. This plant bears fruits and flowers in January and is harvested up to March. For the traditional uses or in the traditional medicines, its dried roots and stem bases are used. And its traditional use is mostly in rasayanas, and it is used to promote health and longevity. It is generally believed, and there are ample evidences, that it augments immunological defense in the body. Lately, like in other, its medicinal properties have also been used and has been witnessed in bronchitis, asthma, ulcers, insomnia. And there is a lot of work which has been done collectively by CMAP and CDRI for its neuroprotective activity. This is to show you that how this herb look like, and its seeds are very tiny, but its roots are very juicy and fleshy. And these are the dried roots which are actually used for the medicinal purposes. Traditionally, it was used in rasayanas, but now they are being repositioned for many other diseases and ailments. Now, what I'm trying to show you that there are large number of biological activities which have been reported in the literature. And if you give a keyword of Withania somnifera, actually you pick up more than 2,500 papers. And it has been very effectively shown to possess anti-diabetic, anxiolytic, adaptogenic, memory-enhancing effects, anti-inflammatory, as was being emphasized earlier, anti-cancer, hepatoprotective, even cardioprotective. And there are reports that it has found to be antiviral as well against some of the viruses. And presently, it is also being evaluated for SARS-CoV-2. Its immunomodulation is well known and well accepted. Its properties such as sedative, hypnotic, hypotensive, respiratory stimulant action, along with bradycardia, and it protects against rheumatism, gout, skin diseases, and it is also well used by geriatric population. In CSIR, CMAP, NBRI, IIIM Jammu, ISCB Kolkata, and CDRI have worked very extensively on this, on Withania somnifera. In fact, CMAP has developed a good number of varieties, and they had been evaluated by various labs for their biological activities. So there are a large number of compounds which are biologically active, have been isolated, and most of them are actually secondary metabolites. A good work has been taken up by NBRI also in this aspect. And there are many phytochemicals which have been characterized. Although there are various chemotypes which have been developed by CMAP, and it was found that they vary in the amount of alkaloids which are present in these, in the roots and the stems of, even in the leaves of Withania plant. So there are some structures which I have actually taken from the literature and have shown in this slide. So vetiverine A, vithanone, vithanolide A, and then ashwagandhanolide, then vithanosides. Then there are many other compounds. More than 64 compounds actually have been isolated so far. As I was mentioning that CMAP had developed several chemotypes, and namely like these three were actually pursued through in the Nimitli project, such as Nimitli 101R, Nimitli 118R, and Nimitli 128R. These, I'm showing you a comparative data of the various compounds which are actually more prevalent in these this plant. They do vary. Some of them are actually present in almost equal amounts, such as vithanol, but they do vary in their distribution in various chemotypes. And the scientists at these institutes have used powdered roots of Withania, and then they were extracted by hydroalcoholic method. And then these extracts were concentrated, lyophilized, and preserved, and then they were evaluated for various activities. As I was telling you that CMAP and CDRI have collectively worked together to establish its neuroprotective activity, and it has been shown to be very protective in both pre as well as post treatment of in the stroke models. So I will be telling you that one of the variety of this Nimitli 101R had been evaluated very extensively for the immunostimulatory effect, antifilarial, anti-leishmanial, and they also showed that it also synergizes with the known anti-leishmanial drugs. Whereas Nimitli 118R has been evaluated and was actually positioned for its neuroprotective effect, and there are large number of studies conducted both at both of these institutions, and it is very extensively evaluated for PK/PD studies by using marker compounds, which are listed, some of them which are listed here. Then its safety pharmacology, regulatory toxicity studies have also been completed, and it is being positioned and to be used for the neuroprotection in the stroke patients. So it is just to briefly tell you that it actually shows a very good protection against the neurological deficit which has been induced by middle cerebral carotid arteries ligation in rats, And it also shows, like there again, like these two bar diagrams or two figures are indicating that it has shown neurological deficit protection as well as protection in the infarct zones. So this significant reduction has actually led to the studies which I had told you before for its safety, toxicity, and also the pharmacokinetics studies. Some of the studies I will like to show you in brief that it also possesses a very good vasorelaxing properties. As you can see that in the endothelial preserved aortic rings, there is a good vasoprotection which was inhibited by NOS inhibition. And then it was evaluated. Then one of the active molecule that is bethanolide was also evaluated for its vasorelaxin properties. And it was observed that it acts through nitric oxide synthase activation. It is not only upregulating the nitric oxide synthase, but also enhances the phosphorylation of eNOS. And this phosphorylation of eNOS actually activates enzymes. So it is acting in both manner. It is inducing its expression as well as it is activating the enzyme because of the phosphorylation. So there was also effects to study whether nitric oxide formation is there or not, which shows that there is significant amount of nitrite accumulation, and it also preserves, because of its antioxidant activity, the tetrahydrobiopterin, which is a redox-sensitive cofactor for nitric oxide synthesis. There are some more additional activities which I will like to touch upon. Here, like in this slide, I have listed, this is a paper which has been published in Journal of Ethanopharmacology. This figure has been taken from there. So if you see in anti-cancer properties, like it on several cell lines, as well as by using various signaling cascade proteins, it is shown to be anti-proliferative. It normalizes the cellular energetics. It stimulates the programmed cell death because it is important to induce cell death through apoptotic phenomena in the cancer cells. It stabilizes the genome, and it also inhibits angiogenesis. So these are the properties which are actually required for any anti-cancer molecule, and it possesses many activities. So we can say that a polypharmacology is being represented in Bethania to exhibit the anti-cancer effects. Not only that, there are some very important immunological activity which has also been seen, and it also inhibits many signaling molecules which have been associated with tumorigenesis, suppression of tumor, and its metastasis. So it's a very busy slide, but I am telling you in brief that it not only inhibits cell growth, it inhibits also metastasis, and it also inhibits angiogenesis and stabilizes the genome. Now, as it was emphasized by Professor Padmanabhan, that immunology component is very important in nutrition as well as in our traditional medicines. And we know that both innate and adaptive immune responses are important. So if you see in this slide, what I have covered is that there are the innate immune responses exhibited by leukocytes like neutrophils in terms of their transmigration, phagocytosis, then signalings. Both are being affected by Bethania as well as its active metabolites. In the adaptive immune response, it affects Th1, Th2, Th17, T receptor signaling pathway, IL-17 signaling pathway, and also in the innate response, it also affects the natural killer cell-mediated cytotoxicity. Interestingly, other cells, blood cells, such as platelet activation is also inhibited. There is inhibition of complement and coagulation cascade. And these are the reason it is being repositioned and for the use in COVID-19. Because in COVID-19, whenever there is a high morbidity and mortality in some of the patients, we see activation, there is a aberrant cytokine storm, There is activation of platelets. Thrombosis is observed. So Vithania could be useful because of its properties. Now, Vithania, as I told you, it is being repurposed for SARS-CoV-2 treatment as well as pathology. So I'll just briefly tell you that this virus is known, like it has been identified that how it enters the cell. And there are two proteins which are very, very important. One is ACE2 and then TMPRSS2, which is a protease. We do have, like we are now repositioning, as well as there are some monoclonal antibodies, as well as convalescent plasma is being used, and they affect the entry. While for the replication, there are several antiviral drugs which have been repositioned, like remdesivir is, I think, showing the highest activity. Then the pH modulation by hydroxychloroquine is also proposed to be effective against SARS-CoV-2 infection. So proteases, entry, and pH, and the viral replications are actually being identified as the possible targets for the new drug discovery, as well as repositioning of the molecules. As was emphasized earlier, that there is a aberrant cytokine storm because of the cells which are involved both in the innate and adaptive immunity. So, but not only that, because the cytokine storm, we get lung epithelial damage, and this is leading to hypoxia and further deterioration in the patients. It also affects the interferon gamma pathway, because interferon gamma are the natural antiviral proteins which are synthesized in our body, but it destabilizes interferon. So we need that how can we stabilize this interferon which can exhibit antiviral activity in the person itself. So the current medical therapy is positioned towards antivirals, and people are also using antibiotics to prevent secondary infections, and the convalescent plasma, which are being used very effectively. So we can divide this disease into 3 stages. One is the initial stage, we have viral response phase. In the second stage, we have like viral response is partly there, but then the host inflammation has also started taking up. In the stage 3 and this is happening at the lung level. But in the stage 3, which is a hyperinflammation stage, there actually other organ systems are getting involved. Like we see kidney failure, we see adverse cardiac events, and pneumonia is actually also worsening during the stage 3. So in the mild stage, we have only mild fevers. Like in the stage 1, we have mild fever, we have dry cough, some people have diarrhea and headache. While in the stage 2, there is shortness of breath. As I told you, the hypoxia starts setting. So there are various clinical signs and parameters which are being monitored, such as lymphopenia is seen in the early stage. There is increase in prothrombin time, and also some people do exhibit some mild change in the D-dimer and LDH. While in the stage 2, we see transaminitis, and which is actually indicating that the patient is entering into the serious stage. And here we see a lot of morbidity, and in the stage 3, we see a lot of mortality, where we see very high levels of cytokines and prothrombotic state. A lot of ferritin is there. So what are the potential existing therapies in this stage? These are antivirals. Hydroxychloroquine was initially used very much, but now there are several clinical trials which are actually challenging the use of this. Then we have remdesivir, which is showing good efficacy in reducing the duration of the disease. Convalescent plasma is also being evaluated. There are trials which are not showing very, very promising outcomes. So what we need is reduction in immunosuppression. So for that, corticosteroids are being used, but we know that when we use corticosteroids, although inflammation is suppressed, but it is not affecting the viral response. So that is also a concern. Various other cytokine inhibitors as well as signaling cascade inhibitors are also being used, and they are in clinical trials. So why are we thinking that Vithania could be useful? Because we know, because of its known effects, it is actually affect lot of inflammatory cells and lymphocytes. And during the severe stage, when we see a lot of cytokine storm, there are reports which are indicative that Withania reduces the cytokine levels, So it is expect, and Withania, as I told you, that there is a potential neuroprotective effect which is already there. It also, because of increase in nitric oxide, is also enhances the cardioprotective roles. So it is possible that the clinical trials which are being undertaken with Withania could be very, very useful and might end up in repositioning Withania for this treatment. So there are, as I was telling you before, and it was also emphasized previously by Suchitra, that these Ayush formulations like Glycyrrhiza, Gynospora, Withania, and Ayush 64 are being propagated, and there are clinical trials which are happening for these formulations. There are already 6 clinical trials which have been registered for Withania, and it's being positioned initially for the early stage treatment or for the prevention, along with like yoga and other booster, Quat and kinase. So I will like to thank everyone for the patience listening. And I want Suchitra to thank you for giving me an opportunity to speak and share my views on. I also want to acknowledge all the CSIR labs who have generated very, very valuable data on Withania. I also like to acknowledge all the researchers who have published its immunosuppression effects, which might be useful for this disease. Thank you so much.
Thank you, Madhuji, for this excellent, so very educative and informative lecture. I'm sure all of you would have enjoyed it. And ashwagandha, which we all know for long as a neuroprotective and full of medicinal properties, now is emerging as a hub from the virus control point of view in these trying times of COVID-19. A promising role of ashwagandha is so very heartening, and as Madhuji told, 6 clinical trials are underway, and I hope and wish that Ayush comes up with some very potential molecules which could be used for treatment of this virus while we all are waiting for a potent vaccine to come up. I think if there are any questions through chat, the coordinator, Dr. Suchitra, or Paramjit ji would take it up at the end of the session. And I move on to the next speaker, who is Dr. Professor Anil Tripathi. Like Madhuji and other speaker too, he needs no introduction, but then just to brief all those who would not be in direct contact, I give a little introduction. Professor Tripathi, presently a professor of biotechnology at BHU, is holding the post of Director, Institute of Science since February 2019, prior to which for 5 years he was Director of Central Institute of Medicinal and Aromatic Plants, CIMAP, at Lucknow. And during his tenure as a director, he provided much desired leadership in spearheading the cause of rural empowerment through cultivation and processing of medicinal and aromatic plants. Under his leadership, CSIR CIMAP was given a CSIR Technology Award in Life Sciences 2015 and CSIR Award for Science and Technology Innovation for Rural Development. As a member of CSIR Society and CSIR governing body, Director General CSIR has also designated him as Mission Director of the CSIR's Aroma Mission. Professor Tripathi, as you all know, is an outstanding researcher. His areas of research interest include bacterial diversities, genetics, functional genomics, and systems biology of Azospirillum brasiliensis for synthetic biology applications. Fellow of INSA, ISC, NASI, National Academy of Agriculture Sciences, NAS, and also Association of Microbiologists of India, that is AMI, and also Biotic Research Society of India, BRSI. Dr. Tripathi is a recipient of several awards, including Professor B.D. Tilak Lecture Award, Professor R.N. Tandon Memorial Lecture Award, Vice Chancellor's Award of Excellence in Research, BHU, member of many committees, many committees for microbiology and immunology, convener of INSA Lucknow chapter. He was also member of several important academic bodies like DBT, DST, and academic bodies of universities. This afternoon, Dr. Tripathi has chosen to speak on potential of essential oils and plant ingredients in preventing the spread of COVID-19 pandemic. Over to Dr. Tripathi, please.
Thank you very much, Professor Veena Tandon. I'm very grateful for NASI for giving me this opportunity. And I see the President of the NASI, Professor G. Padmanabhan, President of INSA, Professor Chairperson, Dr. Manju Sharma'ji, and present and past presidents of the NASI, many distinguished former directors and present directors in CSIR labs, very distinguished scientists, and my previous speakers, the speaker, Professor Madhu Dikshit, and the successive speaker, Dr. Ritu Trivedi. It's a real pleasure to associate myself with this event. And I am grateful to Dr. Suchitra Banerjee for her affection to insist that I must speak in this session. And also I was, given the impression that Dr. Manju Sharma'ji wants me to speak in this session. Although I'm not an expert like Dr. Madhu Dikshit, who had given very, very informative and very, highly technical lecture of the essence of the work of, decades of research, which she has done herself along with her group. But today, what I will be doing is that I'll be, sharing my excitement about some of the recent developments which are taking place with regard to the application of, aromatic plants and medicinal plants and their utility for treating or, preventing the spread of COVID-19. And this is largely because, as Dr. Suchitra Banerjee mentioned, that the Prime Minister has given the phrase the vocal for local. And as has been said by Professor Veena Tandon in my introduction, that I have been batting for CMAP and batting for farmers, or in fact, I should say that I've been batting for farmers through CMAP platform. And today also, I will be doing the same kind of job for batting for the farmers. Dr. Manju Sharma'ji mentioned the importance of establishing Nutri Garden. Actually, some time back, in one of such seminars or webinars, when the National Medicinal Plant Board Chief Executive Officer Dr. Shastri was there, I was also advocating for this kind of concept that every village should have a small primary healthcare garden. So, and the healthcare professionals, some semi-literate people should be educated how to make use of these things. And there are a number of plants, a small number of plants which can be generated or cultivated in these primary health gardens in each village, because not every villager can afford to come to the cities for the secondary treatment, and therefore we need to arrange some facility in the rural areas, in the remote areas, where farmers can take care of their health themselves. And I sometimes feel that the developments in the application of aromatic and medicinal plants, particularly for the health purpose, are not taking place with the pace it is required, because there might be interest of the pharmaceutical companies to promote their own products instead of promoting this kind of local remedies. Of course, local remedies have less science, but they have the tradition of use and their efficacy. But I think we need to implement these local remedies with some amount of science so that they can be better acceptable by the people. So essential oils I will be talking about, as was said about me, that I have been steering this CSR Aroma mission. And through this CSR Aroma mission, I have seen the happiness of farmers when they were benefited by cultivation of these aromatic crops. And I have taken this mission forward up to the international level to International Fragrance Association. I've advocated for many of these essential oils because if they are cultivated by farmers and if they are sold, they are utilized, there will be demand, and then eventually it will result into the farmers' empowerment. And in this slide, you can see that. Yes.
Sorry to interrupt you. Can you make it full screen? Your slides.
Okay. All right.
Yeah.
Okay.
Thank you.
So
thank you.
Yeah, thank you. So here you can see that the essential oils have been in use in all over the world, and in India we say that Sugandhim Pushti Vardhanam. So the aroma, leads to well-being, a feeling of the well-being. But not only the feeling of well-being, it has also been used. The aromatherapy has been used in taking care of many of the health-related ailments like anxiety, depression, irritability, aggressiveness, pain, etc. And a lot of aromatherapy formulations have been in use, and they are quite popular. Not all of them have the scientific basis, but many of them, at least the ingredients, good science has been done to show that why they are yielding the kind of effect which they are showing. There's yet another list of for example, insomnia, the chamomile and sandalwood, patchouli. These are very well proven that they induce the sleep. And there are other essential oils which have been in use, variety of purposes for our well-being. Now, the advantages of essential oils, if you look, they have been known to have anti-inflammatory effects and immunomodulatory effects, bronchodilatory effects, antibacterial, antifungal, antiviral properties. And the important point is that they can reach, their aroma can reach to the places where other chemicals, the liquids, the fluids cannot reach there, but aroma can penetrate all these places. And that is why the volatility of the essential oil is an advantage for their intranasal administration. And the nasal intake of drugs has been shown to improve their bioavailability. And the nasal route has been considered very useful route for some of the drugs which are not easily bioavailable. This issue which has been raised by Professor Padmanabhan. Bioavailability is the main issue, and nasal route is extremely important and being tried for many of the new drugs. And the other point is that this essential oil and their volatiles, they are lipophilic in nature, and they can penetrate the lipid bilayer of the viruses, particularly the envelope virus like coronaviruses. They have lipid envelopes, the lipid bilayer, and direct contact of these volatiles can lead to disruption of the envelope. So there is a possibility that they can be very effective, and they induce positive effects on host respiratory system, which includes bronchodilation and mucolysis. So these, we have been using tulsi for taking the steam for nebulization. We have been using the tulsi and eucalyptus and many other, essential oil producing leaves, and which give a lot of relief. We all know it by our own experience. And they are also constituents of the, the inhalers, which give instant relief. And as the previous, investigations have shown, that the essential oils are very active against a wide variety of viruses, that is like influenza virus, different strains of influenza virus, and herpes virus is also several strains of herpes viruses are there, and yellow fever virus, avian influenza virus, and dengue virus. So the efficacy of these essential oils has been shown in many of the publications, that how they are able to inhibit their growth and multiplication. So yeah. So now here you can see in this picture that the influenza particles, a lot of studies have been done in past on influenza. The epidemiology of the influenza is somewhat similar to that of the COVID or coronaviruses. So the influenza particles, they spread by airborne droplets, which you call fomites, and through close contact with an infected person and contact with objects contaminated with the virus. You can see on this right-hand side, the person is sneezing, and because of that, a lot of fomites, they get spread in the air, and they stay there for quite some time. And there, the spray of these essential oils or their vapors or their volatiles can be quite effective. And there are observations which I'll be elaborating further, that they can neutralize many of these fomites which are present in the air. The influenza particles, they remain viable on objects for up to about 48 hours. And this is the data of the CDC. So effective sterilization of objects is an important step in preventing this influenza. And the property of the diffusion of essential oils is considered very effective in eliminating the airborne bacteria and viruses. And that is, to do that, the nebulization of essential oils of blue mallee, that is called Eucalyptus polybractea, or tea tree, that is Melaleuca alternifolia, just for 15 seconds can effectively and rapidly eliminate airborne influenza droplets within 15 minutes. So you can see the efficacy of inactivation of the airborne influenza virus has been proven by these essential oils. Now, going to the next. It is not moving. Yeah. So when we talk about the mechanism of action of essential oils, they are not studied as much in depth as you have heard in the previous talks, but there are some prima facie studies are there, prima facie evidences are there that they can be effective. For example, the treatment of the influenza virus with lemon balm oil, this prevents attachment of influenza virus to the host cell surface, cell receptors in vitro. And similarly, the tea tree oil, which I have mentioned, this prevents intracellular processing of the viral particle. When introduced into the cell culture media, this tea tree oil prevents viral uncoating by interfering with the acidification of the endosomes and membrane fusion. And there are many reports which have already been published. You can see the pictures of these plants. On the left-hand side, you see the lemon balm, and then you see these tea tree. And then also you see the eucalyptus, which have been found very effective, and some of the part of the mechanism is understood. Here you see that the simple diffusion, by a simple diffusion of essential oil, that can have considerable reduction in the influenza in the surroundings. And you can see here the plants which are this bergamot, that is the Citrus bergamia, and these are the Cinnamomum gelanicum leaves are there, and then again eucalyptus and geranium. The oil, the aroma of these oils, this is able to reduce this fomites. The passive diffusion of this bergamot and blue gum essential oils for 10 minutes, or cinnamon leaf or geranium essential oil for 30 minutes is considered enough to reduce influenza activity in cell culture. Not only in the environment, but also in the cell culture, this can be done. Now, here are some data which are taken from some of the publications. The list of the oils like tea tree, blue valley, bergamot, blue gum, geranium, cinnamon leaf, clove buds, cassia, and they are effective in killing the airborne influenza viruses on the surfaces, and they can prevent pneumonia, and they modulate also the immune response to influenza. I will not go into the details, but some directions are given that how much exposure to this essential oil volatiles can be effective enough. Now, in the next slide, you can see that there are some essential oil blends which have been made and which have been found to be very effective against influenza virus. And in fact, this blend 2, if you see, this is a commercial blend which contains cinnamon bark, clove bud, sweet orange, blue gum, and rosemary oils, which effectively check growth of influenza virus in cell culture. So it's not only preventing in the air, but also they show efficacy in the cell culture. But they have not been taken forward, and I think there is a need for taking them forward in the in vivo systems and also clinical trial. Now, in recent time, when you go to some of the doctors which have some trust in the traditional medicine, they always say eat a lot of garlic, and garlic oil has been shown to have some evidences that they can be very useful, and garlic has been traditionally used for treating cold, influenza, and other infections, and their main constituents are several. They include mainly the allyl disulfide, which is about 28%, allyl trisulfide is about 22%, and then allyl propenyl disulfide, allyl methyl trisulfide, and diallyl tetrasulfide. So there are about 18 ingredients which are in prominence, 17 ingredients of garlic essential oil. All of them show binding with the host protein, that is the ACE2, as well as with the viral proteases, indicating great potential of garlic oil in treating the COVID-19 patients. But mind you, this is only in silico binding, which is a very, very preliminary indication that these ingredients can be quite effective. The treatment of primary human pulmonary artery endothelial cells with garlic oil ingredient, they downregulated about 36 genes, which control the expression of majority of cytokines identified in the cytokine storm, which was discussed by Professor Padmanabhan and Dr. Madhu Dikshit. That happens in the severe cases of COVID-19. So there's some evidence that it can mitigate the effect of the cytokine storm caused by the COVID-19. And the diallyl sulfide is known to induce the NRF2 activation in the lung MRC5 cells, which is suggested to prevent oxidative stress induced during the lung injury. And this has DS, diallyl sulfide, also has the potential to prevent the entry of COVID-19 into host cells, as well as activate molecular antioxidant pathways to decrease secretion of pro-inflammatory cytokines. So these are some of the literature which actually which I tried to collect, that why the garlic has been recommended. And you can see that there is also analysis of the garlic oil which has been published, and you can see that these are the about 18 ingredients, and out of these 18 ingredients, about 17 of them they find to different proteins which are involved in the viral infection. Now, again, here there is some evidence on the basis of in silico studies, some in silico publications, that the ingredients, they can bind to the angiotensin converting enzyme 2, and there are at least 4 of these ingredients which show quite good binding to this angiotensin converting enzyme 2. And also, there are ingredients which are able to bind to the main protease. So that is an indication that the garlic oil, there is in silico at least there is a potential which is showing that this can, be a good natural, support for controlling the COVID spread. Now, eucalyptus has been used for quite some time. This is it's a main ingredient. It has been used for pharyngitis, bronchitis, and sinusitis, and its main ingredient that is called 1,8-cineol, and it is also called as eucalyptol. This is the major ingredient of the Vicks VapoRub, which most of us have used or we have been using, and it acts as a nasal decongestant and has a profound bronchial anti-inflammatory activity. This 1,8-cineol exhibits muscle relaxant effects also by decreasing smooth muscle contraction, and the clinical studies have been done which show that inhalation of 1,8-cineol exerts anti-inflammatory and analgesic effects, and therefore it can be used in bronchitis, COPD, and asthmatic patients. The eucalyptus oil and 1,8-cineol show antiviral activities as well, and that is against enveloped mumps virus, herpes viruses, and the influenza virus. And again, some of these observations, like binding of the gensinone and 1,8-cineol, has been shown through molecular docking with the main protease. And the treatment with the eucalyptus oil and 1,8-cineol reduces the release of pro-inflammatory cytokines from monocytes and macrophages. Now, with these evidences, there has been an over-enthusiasm on the part of the aromatherapy industry. And there were at least 7 aromatherapy industries which were given a warning letter from the FDA. And this is one of the warning letter issued to a quintessence aromatherapy company. It is not because there was any adverse effect or so, but I tried to go deeper into it, that why the warning was issued. The warning was issued because they had not done enough experiments or experimental evidence to show that these formulations or these blends of essential oils, they are actually effective against COVID-19. So they made certain claims, or they at least projected its potential, but I think this was not accepted to FDA. And this is the statement of the quintessence company. They say that the wide range of essential oils have been clinically proven to possess antiviral properties, and they have given examples of several influenza virus strains, parainfluenza strains, vaccinia strains, and herpes simplex and polio viruses. So these essential oils have been found to be effective. In fact, some of these essential oils have been shown to be effective against the coronavirus epidemic, which was there in 2002-2003 in China, and this viruses were inhibited by some of these essential oils, and they have recommended that some of the most powerful antivirus essential oils with potential to provide defense against coronavirus, they include some of these basil, bergamot, cajupot, cedarwood, etc. But the point was that they made a recommendation based on previous observation against many other viruses, but they did not have evidence against the COVID-19. And I think there is a need. I have advocated. Dr. Prabodh Trivedi is sitting here, and when about 6-7 months ago, I had a discussion with him, and I suggested that CMAP being the torchbearer of essential oils, we should take up this kind of studies, at least some in vitro studies in collaboration, to at least clearly show that some of the essential oils, they have the capability to inhibit the growth of or the spread of the infection of coronavirus COVID-19. So again, there was some further claim, which is some examples of claims. Sorry. for intended use of, this is the part of the warning letter of the FDA, that some examples of the claims on your websites that establish the intended use of your products and misleadingly present them as safe and or effective for the treatment or prevention of COVID-19 include the following. So they have given some of the statements which are not very scientifically proven. And of course, I would agree that there is an institute in US which is called Research Institute on Fragrant Material. It has been testing the allergenicity and the sensitivity of some of these essential oils. And many of these essential oils can create allergic responses. And therefore, I know this is related to the respiratory disorder. And if it is inhaled, it is possible that to some patients, some people, it may lead to severe allergies. And therefore, one has to optimize these things. Of course, FDA has issued a warning for this, but I think there is a lot of potential in testing each of these essential oils individually, because there is a possibility of finding some solution for preventing the spread. Now I will shift my gear to the plant which is called Artemisia annua, which is a favorite plant for CSIR, and particularly for CDRI and CMAP, which have made a lot of contribution in developing this anti-malarial, and the National Malaria Research Center also, the ICMR, they have all contributed together in promoting the use of the Artemisia related compounds, the artemisinin and its derivative alpha-beta artether. And this Artemisia has not only efficacy in, I know this plant is also very dear to Professor Padmanabhan. So this plant has not only potential against malaria, it has also been shown to be effective in some kind of cancers as well. And in 2002 and 2003 epidemic, this plant was found to be effective against this SARS-CoV-1 spread in China. The Chinese researchers in 2005, they have shown the efficacy of Artemisia annua in extracts in containing its spread. And the pulmonary fibrosis, which is observed in the SARS coronavirus infection, which increased in severity mediated by interleukin-1. And I think the Artemisia annua extract has been shown there is some evidence that it prevents this fibrosis. And in fact, one of the derivatives of Artemisia annua, which is artesunate, it is a promising novel drug to treat pulmonary fibrosis, as it is able to inhibit pro-fibrotic molecules associated with pulmonary fibrosis. So here is the president of the Madagascar, the Regulina, who promoted a herbal blend made from Artemisia annua, which is called COVID Organics. It has a major component as Artemisia annua. And this is a kind of very highly unscientific gesture on the part of the president to show that this drink can be very, very effective, because there was no scientific evidence. And in fact, the WHO has urged extreme caution over these kind of reports which talk about the efficacy of such products. And WHO has warned that there is no evidence to suggest that COVID-19 can be prevented or treated with this. Of course, this is there, but there is some promise which was further substantiated by a group in Germany with the Max Planck Institute. And here, Professor C. Berger, who is the director of the Max Planck Institute for Colloids in Germany, he has shown, after months of studies, they have been able to show that Artemisia annua extract is quite effective, and when it is mixed with the coffee, it can be very effective. So that is why this I got it from the net. Does Artemisia annua offer a cure for COVID-19? If that is the case, then the answer is that buy Professor C. Berger's Artemi tea to survive COVID-19. This is still, a lot of studies are going on. This is again Professor C. Berger. It says that after months of analysis, this Max Planck Institute in Potsdam in Germany announced that Artemisia annua extract has been shown to be very effective against coronavirus, and further clinical trials will be launched. This is the news of the May 2020, and then these clinical trials have already been initiated in United States and Mexico. And the Artemisia annua leaf extract was shown to be to have significant antiviral activity against coronavirus after being extracted with pure ethanol or distilled water, even distilled water. And antiviral activity increased significantly when ethanol extract was combined with coffee. This observation, attracts me a lot, because these simple methods of growing an Artemisia plant, making some simple extraction and using it, provided it can be scientifically proven, this can go a long way in the primary healthcare system of our country. That is why I look at this news with a lot of excitement, because not that some specific drug can be developed which can go to the elite classes, but that our rural masses can make use of it for health benefits. And this is some detail of the clinical trial, where about 360 participants have been registered, and the randomized placebo-controlled multi-center, multi-arm clinical trial is being done in Mexico. And these are some details that it will be completed in January 2021, and the primary completion date, and the study will be completed in February 2021, and the trial has just started 2 months, last month in September. So this clinical trial, the randomized clinical trial, the hypothesis is that the addition of the agents that inhibit viral entry or replication of SARS-CoV-2 virus, such as Artemisia annua and chemostat. They have kept chemostat, about which Professor Madhu Dixit also discussed in her talk. So this will reduce the rate of a composite outcome of hospitalization due to COVID-19 pneumonia or the use of oxygen therapy, and will be devoid of additional moderate to severe toxicities, because Artemisia extract have been thoroughly tested for the safety, and they have been found to be highly safe. And the expectation is that the clinical outcomes in COVID-19 infected patients at higher risk of poor outcomes following infection will be improved compared to the standard of care when introduced as an early intervention after diagnosis. Now, I would not go into the details about the Ayush Path, that Kadha, which has been already mentioned, and the Tulsi, Dalchini, Sunt, the Adrak, and Kali Mirch. These are very simple, the kitchen ingredients which can be used, and in fact they have been probably very, very effective. And some prima facie studies, particularly with the in silico studies, which are not considered very scientific, but they are of course suggestive that some experimental studies can be done. And here you can see that I think this kind of slide has already been shown by Dr. Madhu Dixit. But some of these ingredients of this phytochemicals which are found in the kadha, they have been found to affect the spike protein. They bind to spike protein. They affect the virus entry, and they prevent inflammatory response, and they also affect the uncoating of the virus and completion of the viral life cycle. And there are some results which have been shown by some BRC scientists that through the docking approach, that many of these ingredients which are present in this kadha, like somniferine, closporide, and flavonol, vithinolide, epigenin, they all are binding at the same place in this main protease of SARS COVID-19. Now, there are some other examples like selected phytochemicals which are superimposed here. You can see that vitafrin, vithinolide B, Dr. Madhu Dixit has already talked about it. And there are other compounds which are showing binding to this ACE2 protein. And here is yet another example of the binding of some other herbal ingredients which are able to bind. I think a lot of studies they need to be done, at least to begin with some of the plants which are grown in India, some of the essential oil yielding plants, some of the, like Artemisia annua, which has shown a lot of promise, a lot of research has been shown. And these plants, I think, need to be investigated deeper. And if we go further, I would like to just go through this statement of the Prime Minister that evidence-based herbal pharmaceuticals and nutraceuticals is a national call. And Prime Minister has said in 2016 in one of the meetings of the Ayush that we must apply the techniques and methods of modern science to test and validate the results of our traditional knowledge and principles of Ayush to assure quality of our products and health services. So I would like to end here and thank everyone who has been hearing this talk, and to NASI for inviting me to share my sentiments. I have accepted the call, but I know that this is not my domain. I have only tried to share my excitement about the new developments. Very much.
Thank you so much, Dr. Tripathi, for your exciting and full of information lecture. Of course, many of us knew many things about the essential oils which are routinely used for malish and for common ailments like cough and cold. But then knowing so much in detail about their potential role as phytotherapeutic agents against COVID-19 seem very exciting and promising. Thank you so much once again. Now, I would like to move on to the third and the last lecture in this session. And the speaker is Dr. Ritu Trivedi. Dr. Trivedi is presently working as a principal scientist and associate professor of ACSIR Endocrinology Division of CDRI Lucknow. After receiving her PhD degree in the year 2000 from SGPGI Lucknow, Dr. Trivedi did her postdoctoral fellowship from National Institute of Health, Bethesda, USA. Her research has been focused on metabolic bone disorders, specifically on osteoporosis and osteoarthritis. She has worked on several molecules derived from either natural resources or chemically synthesized for improvement of bone health. She has worked on the standardized fraction of Dalbergia siso particularly, that acts as an osteogenic agent, which has led to the translational benefits as a rapid fracture healing agent, as well as is valuable in primary osteoporosis. Her work on nanoformulation of Spinacia auricea has led to the product for osteoarthritis. These technologies have been licensed by Parmenza Herbals Private Limited for postmenopausal osteoporosis and osteoarthritis, and both are in the market by the name Reunion and Joint Fresh. In addition to translational work, Dr. Trivedi has established involvement of microRNA's bone formation and has shown how miRNA exerts skeletal anabolic effects epigenetically during bone formation period. With more than 100 research papers in journals of repute and patented few technology, Dr. Trivedi has co-authored several book chapters, made more than 50 invited lectures. She is executive member of Indian Society for Bone and Mineral Research, is a recipient of prestigious Jyotia Memorial Oration Award, Tata Innovation Award 2019 by the Department of Biotechnology, P. Shield Award 2019 by NASI. She is, by the way, also a fellow of NASI. Her contributions have led her to be designated as an important member of the thematic group health initiated by the Office of Principal Scientific Advisor, Government of India, and DST. The group is in the process of formulating India's new science, technology, and innovation policy, that is STIP 2020. At institutional level, she is coordinating research and development activities of the thrust area, bone health, and phytopharmaceutical mission. This afternoon, Dr. Trivedi would be telling us about the musculoskeletal health in the times of COVID. I call upon Dr. Trivedi to start her lecture, please.
Thank you, ma'am. And I'm really grateful of what you've described me. And I hope I do justice of what you just told about me. And good afternoon to everyone and very distinguished people present in today's webinar series. And out of these, some of them have been leading directors of
Not very audible, no? Is it my problem? Can the volume be increased? Is it possible?
just see. Hello?
Dr. Trivedi, just
Am I audible now? Hello? Hello?
Yes, you're audible now. Please speak a bit loud.
And my
Speak
slides?
Dr. Trivedi?
Yes, hello?
Yes, you are audible now.
Okay. And can my slides be seen? Hello?
Ritu, you have two computers, Shraddha's and Ritu Trivedi. I think you have to shut down one and then just present from one.
Hello? Can my voice be heard?
No, you are not still audible clearly. Please close the other login point and speak from one only.
या आप सर श्रद्धा सिन्हा नाम के कंप्यूटर को आप उसको अनम्यूट कर दीजिए. The voice will be audible. Hello?
Absolutely.
Hello. Yes. Your sharing permission is at the login point of Shraddha Sinha, and
Yeah,
you
yes,
can speak from yourself, but sharing permission has been given to Shraddha Sinha.
and also, yeah, okay, okay. Can slides be seen now?
Yes, slides are visible now. You please start.
Yeah, yeah, okay. Thank you so much, and sorry for this glitch that has happened. And I'm really, really very thankful to the organizers for having me here, especially Suchitra ji, who's been running up and down and making it possible. Uh, the chairperson, Dr. Padmanabhan, and I have my own directors who've headed CDRI, part of NASI fellows, and speaking on this platform today in this webinar. Dr. Madhu Dikshit, Dr. Kamboj is there, and in fact, even Dr. Alok Dhawan for some time. And very, very affectionate Dr. Tripathi, who just spoke before me. And NASI has done a commendable job of
Dr. Rituji, please speak aloud. Please.
hello.
Please speak in a louder.
Okay. NASI has done a commendable job by putting bioresources for combating COVID-19. And I am not a plant scientist, but what I've done is in the past 10 years of what I've been working in plant sciences, I've tried to put all the data together and tried to make sense of what SARS-CoV-2 has done to us. So I'll be speaking about musculoskeletal disorders in this context. So, this musculoskeletal disorders has been explained by Darcy long time back in 1917, where he put a very, very strong argument by saying that muscles and bones are inseparable and they're associated and connected with each other. They mold into each other and come together. And this was put by Darcy in 1917, where he wanted to put the this biological phenomenon he wanted to explain in context of mathematical terms. And this theory put by Darcy in 1917 is even being explained today, where by very, very simple experiments where we try to see that really bone and muscles are associated with each other. Bone fractures that are covered with muscle fibers, they heal very, very quickly. This is a lot of data from my lab, and others have also shown that if you have a fracture and if it is covered with a muscle flap, it will heal very quickly, as opposed to a fracture that does not have a muscle flap over it. Then another way of showing that both bone and muscle are associated with each other, the if you block the porous barriers between bone and muscle, they inhibit healing, and because the peptides and the growth factors cannot pass through the pores. And in the same way, it has been shown that if somehow muscle atrophy occurs, or there is muscle wasting happening, then it is also associated with bone loss. Having said this, as I said, that my experience with plant sciences has been in the past 10 years, and I'm really, really grateful to Dr. Rakesh Maurya, who introduced me to this world of plants. And I began my work in plant-derived resources by beginning with Delbergias tissu. And this Delbergias tissu belongs to family Fabaceae, and it just grows throughout the sub-Himalayan tract. Not only the sub-Himalayan tract, you will find this tree by the name of rosewood or teak even growing along the roadside. And what we've done is that the renewable part of the plant has been used in this study, and we've shown the oral human consumption is already known for it, and medicinal purpose for which it is used is known. And what we've done is we've used the renewable part of the plant over here, basically a tree, not even a plant. So after going through 53 plants, screening 53 plants, we found that Delbergia sissu actually had bone anabolic effect. Not only did it show this property, but um, we were very lucky to find a novel and an abundantly present biomarker in it by the name cavionin glycopyroxide, which we have abbreviated as CAFG, and this led us file a patent, which has also been granted now. We've shown its effect in postmenopausal conditions, and the reason why I bring these studies is that I'll show you later in my slides that you have an inflammatory response when you are postmenopausal in condition, or when I touch upon the other diseases also. And we've shown that how Delbergia was osteogenic and helped in bone formation. Not only did it help in bone formation, but it was not estrogenic. And it is very, very important that any of these the plant derived molecules that we have, they are not toxic, because sometimes phytoestrogens, as they are known, they have side effects or the toxic effects on the uterus and the breast cancer. They cause even breast cancer actually. So for long time therapies, it is very, very important that they are not estrogenic, and we found that Delbergia was not estrogenic. We went ahead to do studies on fractures and see that whether it could regenerate bone. And we found that not only that it could regenerate bone, but the amount or the quantity as less as 1 milligram per kg of CAFG was effective in treating the fracture sites, or the callus was formed as the fracture site, and a person regained bone strength. So this is the not only the importance, this is how potent these molecules could be. And as Dr. Padmanabhan said, that there, and even Dr. Tripathi said, that the need of the bar is that we have evidence-based scientific evidences to show that these are backed by mechanisms of how they work. We went ahead to see how CAFG molecule works, and we've seen that this molecule targets the Wnt signaling pathway. It phosphorylates GSK3 beta and lets beta catenin go into the nucleus, and this is how osteogenic action happens. Not only this, and this is on the right side of your screen, you will see that as opposed, we worked out the entire small steps that take place in the beta catenin pathway to show how it inhibits the other inhibitors that are present in this pathway to let beta catenin translocate into the nucleus. And this is the translocation into the nucleus, and it imparts its effect, because of which we are seeing fracture healing taking place. Not only does it target the Wnt catenin pathway, but you'll also see the BMP pathway being affected. And if you go to the treatment and see, there is the recombinant BMP2 is in the market for treating fractures, for treating complex fractures, but it is tremendously expensive. So CAFG could also target the BMP2 pathway, phosphorylate the SMADs, and then in the similar way, enter into the nucleus and show osteogenic response. And this is at the protein level, we've seen how CAFG functions. And the third way, how it imparts its anabolic response or bone forming ability, is that to shut down the cells that resorb the bones and let the bones that form the bone, that is the osteoblast cells, keep letting them form the bone, and don't let osteoclast that resorb the bone to differentiate and kill the cells. So this is the mechanism of how CFG molecule works. This is the product that has come out for fracture healing in the market, as ma'am just described in my introduction. We've done a 1-year clinical trial in postmenopausal women, the paper of which is also published and is in the public domain, where we have shown that in postmenopausal women, it does not allow bone to deteriorate further. It does not allow bone mineral density to go down of what they are detected in the postmenopausal state. So it is in the market by the name Reunion. The patent of it is granted and obviously commercialized by now. And Iris is marketing, and it's also available on Amazon. It was happy to see the lay press covering our work, and it builds in you an enthusiasm of working further. And it was at this time only that I came across Spinacia oleracea, which is also known as palak, and the effectiveness and the efficacy of it that I will show you, what in Hindi we called is the desi palak. And we've compared the two, and we've shown that it's the desi palak that is more effective as opposed to the other palak or the spinacia that is present in the market. So, I mean, it is an area of unmet medical need, osteoarthritis. Why? Because you have no treatments whatsoever. The only treatment that is there are the pain relievers, and they are generic and they are non-pharmacological approaches. People tell you to exercise, lifestyle changes, and pharmacological treatments involve the non-steroidal anti-inflammatory drugs, which are also known as the NSAIDs or the COX-2 inhibitors that are there in the market. Now, in terms of in India, if you see the landscape of how osteoarthritis is affecting, you will see like 22% to 39% people in India are affected by osteoarthritis. And 45% of women above the age of 65 years experience osteoarthritic conditions, and 70% of the women above 65 years have radiological evidences. So you can see the impact that it has in India at the moment. Again, if you'll see the first place, this is how your joints look like in the normal conditions. But if you are osteoarthritic, you see that shiny glaze of the cartilage that is present on the condyles is lost. You have bony spurs on it, which are known as osteophytes. If you go to the doctor and show your X-rays, they will tell you that osteophyte formation ho gaya hai, and now treatment is required for osteoarthritis. Then I'll go to next slide, and I'll show you that how we've gone ahead to show that this architecture that you see, we've created an ACLT model for it, which is a human mimic model, and you see how the regularity of it is lost. And when we give spinacia at 125 and 250 milligram per kg body weight doses, see this comes back with as opposed to what you see in ACLT. We went ahead and we've done histological sections of it, and we've seen that the proteoglycan covering that is over there is so lost in over there, and we've done it also with the taurodine staining. Okay, so there are various scores of looking if you are getting well from osteoarthritis or not, and one of them is the ORC score, that is based on how your knee histology looks like, how bad you have been affected by osteoarthritis, and on the basis of those scores, so the lower the score, the better it is. The lower the ORC score, that means the better your joint is getting, and that is what we see with spinacia, that the ORC score goes down. We did some behavioral experiments to see does it relieve in pain, and yes, it does relieve in pain. If you see the distance traveled and the walking ability, it becomes better with spinacia. So this formulation, when tested at the gene expression level, we see the transcription factor SOX9 gaining up and agrican coming back again, as opposed to what you see in the osteoarthritic condition. And this we have been able to license it to the partner Farmenza, and it's in the market by the name Joint Fresh, and it is again available on Amazon also. So after studying these two, working on postmenopausal osteoporosis, fractures and osteoarthritis, I also came upon another disease that is obesity. And I think Dr. Padmanabhan was mentioning in his introductory session that it is becoming epidemic in India. Obesity is epidemic in India, and it has grave consequences. And as much as you read about it, once this pandemic has hit us, I'll show you data that how bad it could be if you're obese and you are affected by SARS-CoV-2. So these are animals that are high fat diet fed, and you can see in the peritoneum that there's a lot of fat inside. And this is how, if you do a scanning of an animal, you'll see the intraperitoneal, the subcutaneous fat present in the animal. And if your BMI index crosses 22.5, that is in India, you are in the category of being obese. And when you are this, then the fat does not retain itself into the peritoneum, but also goes into your bones. And once it's in the bones, the yellow thing that you see over here, this all fat in the bones. And the lower panel shows you how you're affected if you have fat in your muscles. And what happens is, when this fat is there in your bones, the vascularity, the vascular capacity of your bones decreases. If it's in the muscles, you feel lethargic, you feel sleepy, because so much of fat is there, and constant glucose consumption is happening, not to keep you healthy, but it's for the fat cells that are present in the muscles and the bones. Okay. So as I said, that International Osteoporosis Foundation has said that by 2035, 4 out of 10 people are going to be obese. And this is the world data that I'm showing you. We did simple experiments to see, very, very simple experiments to see that how obesity differentially affects males and females. And we got very, very exciting data showing that if you are adult, if you are an adult female or an adult male, then we see that it is the males that are affected more as compared to the females. And this was really exciting for us to see that you see a glucose intolerance pronounced in females, but males show more deterioration of the bones. And this is what I show over here in the graphs. But as you age, and we did this differential study in the males and the females, and in the females, when the menopause strikes you, and as males age, then it's a dual insult in the females that you're aging and you're menopause, in a menopause condition where estrogen goes down, then this disease deterioration increases in the females as compared to the males. And this was really, really exciting data for us. In fact, we also found that in the males,
Neerajji, have we lost contact?
Yes, no voice coming.
Hello?
Rituji, yes,
Hello?
yes, now again, yes, please.
Okay, sir. So when we question ourselves that what does this obesogenic microenvironment do to us, what happens is that if you're obese, then obviously you have chronically high leptin circulating levels, which is a pro-inflammatory cytokine, as opposed to anti-inflammatory adipokine, that is adiponectin. You become insulin resistant, and you are more prone to diabetic conditions. In fact, you have high concentrations of several pro-inflammatory cytokines, the TNF, MCP1, and IL-6. What do we do about this? Hello? Am I audible?
Now you're audible, but your slide is not visible. Earlier, your slide was visible and you were not audible.
Now is the slide there?
Not yet. No. The slide is not visible.
Just a moment.
Dr. Rituji, please. Shraddha Sinha ji can display the slide, but Rituji, you will have to speak from your own end.
Please let me know if the slide is there now.
Slide is not visible.
Not yet.
Huh? Not yet?
No.
From my computer, I can see the slide.
Share
But you have
button.
to share it. Please share it.
Yeah, it's shared.
No, we can't see it. So when you must have unmuted, you must have clicked some of the button.
It would be better if Shraddha Sinha ji will log out from her end. Perhaps there is interference. So Shraddha Sinha may log out, then it would be audible as well as visible.
I log out, you say?
Yes.
I'm sorry, just give me a moment.
Rituji, now you can present on your own.
Okay.
Yes.
Okay. So what we've done about this, we've been working on ashwagandha and commonly known as
now the slide is coming.
Commonly known as ashwagandha, and I think ma'am and sir, both of them just summarized of what it has been shown, but I will go ahead. Please let me know if my slides appear.
Yes, now the slides are visible.
Visible.
Okay.
Put it on full screen.
Yes.
Yeah. Okay, so we've been working on ashwagandha, and we show that so many products have actually come out of ashwagandha research. And as ma'am said earlier in her presentation, that's an Indian ginseng, and it is actually impossible to take away the credit of this the use of Vidania somnifera, which is an exceptionally good molecule, plant, and the molecule it contains. So if you see the chemistry of this ashwagandha, you will see that it is the 24-methylene cholesterol, which is an important molecule. And every time it can give you like 200 phytochemicals from 20-methylene cholesterol, just depending upon how the hydroxylation changes. If you have 27 hydroxylation happening, you get vidoflurin A. If you have C17 hydroxylation happening, it is vidanone. And if it is C20 hydroxylation happening, you have vidanolide being formed. So if you look at the chemistry of 24-methylene cholesterol-driven molecules from Vidania somnifera, so many uses have been shown, and since they have been summarized before by my earlier speakers, I will not put a lot of time into it. And what we have seen is that Vidania somnifera enriched extract at very, very low concentration is very effective not only on the bone, but it also decreases the body fat. at different doses we've done, and we've shown that it decreases body fat. So when I put all these things together, and when we see COVID-19 striking us, it is true that COVID-19, we cannot deny the fact that COVID-19 is a respiratory disorder, and people usually in close contact do get COVID-19. It's just a debate about how much it stays in the air. What is the particle size? 5 micrometer particle size, lesser size, bigger size, we don't know. The research is just happening, and we are still getting new information every day. It strikes the infected surfaces, the stool, the blood, the semen, and ocular secretions. Domesticated animals are affected, not affected, still a question with COVID-19. And it spreads by coughing. And why do we see such a landscape of COVID-19 affected so many organs is because it goes into intracellular vesicle trafficking. It's found there. Ubiquitin ligases, it functions to inflammatory signaling, nuclear transport, cytoskeletal stability, and mitochondrial respiration also. So if you look all the data that I showed you just now, and if you think about SARS that happened some time back, these were the pressing questions even then. And that was that myalgia, or the pain that was happening in the muscular pain, was reported by 50% of the patients. And when the patients recovered from COVID-19, they showed decreased strength and endurance. And so much so that even the joint pains happened in so many patients. And now that we are getting autopsies of COVID-19 patients, we see there is so much of muscle destruction also along with it. Now the question is that why do obese patients present worse clinical picture than the lean subjects is based on the chronic inflammation they represent. There is reduced macrophage activation, blunted pro-inflammatory cytokine production, and this reduced macrophage activation after an antigen is presented explains that they may have poor vaccination success in obesity, in the condition of obesity. So what is CSIR doing in this regard? I think it has been summarized by Dr. Madhu Dixit also in her presentation, but there are 5 verticals under which CSIR is working, be it surveillance, including digital methods, rapid and cheap diagnosis, in which we have the Feluda kit coming, which works on the CRISPR technology, development of new therapies that includes repurposing of drugs, and it is on this point that a lot of bioresource research has been done and clinical trials executed. Then you have the hospital assistive devices and the supply chain and the logistics. In terms of bioresources, if you see, then Dr. Tripathi showed in his presentation that chloroquine, yes, derived from Cinchona officinalis, shows potent treatment for COVID-19 patients. He's gone up to show some of the clinical trial data also. Then again, you have Tamiflu or oseltamivir, a drug developed for treating influenza viruses, H1N1 flu viruses. And Roche actually started selling oseltamivir under the trade name Tamiflu. Now, it is very interesting to see that oseltamivir was actually discovered by scientists at Gilead scientists using the shikimic acid as a starting point. And it is from this, on the right side that you see is the Chinese star anise, which is a spice from which they derived this molecule. And because these molecules obviously are present in lesser quantity, they've gone ahead to do, they've manufactured it recombinantly using E. coli in the year 2006. So, I mean, if you see the metabolic pathway of how they work, the secondary metabolism is totally dependent on how carbon fixation happens in the primary metabolite. And if the through primary metabolism, you have aliphatic amino acids being formed, then shikimic acid pathway uses this, and it is used to form aromatic amino acids, which are the basic building blocks of protein synthesis. Then shikimic acid it is a 9-step process. This is how oseltamivir was synthesized. They've also shown, studies have also shown that shikimic acid is responsible or used for chloroquine and hydroxychloroquine that we've been hearing in COVID-19 for so much of time now. And what it does is that it is in the, it's by lowering the pH that the release of the virus is inhibited, and this strategy is being used for treating COVID-19 patients. Okay, the question obviously comes that if I talk about musculoskeletal disorders, then is COVID-19, how does it affect the skeletal muscles? Then the studies show that the ACE2 is present in the skeletal muscles. It has also been found in the synovium, the bone, the cartilage, and the meniscus. And this is how the COVID-19, the arthralgia that I explained, or the myalgia that I was showing you, is because this enzyme has been found in the skeletal muscles, synovium, and the cartilage. How does it affect the pulmonary inflammation happening by TNF-alpha and the others that you see affects the skeletal muscle and the bone and the joints, and this causes the myalgia, the bone pain and the muscle pains, causes fatigue and the weakness that happens when you're affected, the primary symptom that you see when you're affected by COVID-19. Now, UK has done a clinical trial showing dexamethasone for COVID-19, and we've got some of our own experiments showing that long-term of dexamethasone is not very good for the body. It allows muscle wasting happening over here, and the muscle weight decreases. And if you see the cross-section area over here, the vascularity of the muscles also decreases on long-term taking dexamethasone for if at all given for. This is a recovery trial that has been done in UK to show the effect of dexamethasone. It shows muscle wasting. You see the muscles, the fibers, the protein synthesis is badly affected as opposed to the control, and this is how it harms the muscle cells. Now, if I put all my data together to see that what conclusions can I draw of these studies, then menopause or ovariectomy that we do where we cut down the estrogen in the body is an inflammatory response. And what we found is that out of the many, we cannot deny the credit of Vidaferin A, that it is very, very effective. It decreases the inflammatory response, the circulating levels of TNF-alpha. Even in vivo conditions, it has shown that TNF-alpha levels are decreased with Vidaferin. Even it is much better than when we compare it to the standard drug of care that are there in the market. It is also effective in reducing the levels of IL-6 and the MCP1, which we talk about. We've talked about the cytokine storm in COVID-19 situations, suggesting that Vidaferin A is the molecule of the day. It inhibits the TNF-alpha induced nuclear translocation via the NF-kappa B pathway. And if you'll see in the end, it does not allow it to go inside the nucleus. So it acts as a very, very good inhibitor. In the osteoarthritic conditions, if you see, Vidaferin has shown very good effects, both IL-1 beta, which is destructive, or whose levels, IL-1 beta and the TNF-alpha levels are increased. You'll find in the MIA is the condition monosodium iodoacetate, by which we create an osteoarthritic condition, and you find these levels so much hugely increased, and Vidaferin A is able to bring them down as opposed to the osteoarthritic condition in the joints. What do I conclude from these slides? That if you see the binding assays that have been performed and what we've done in our capacity also, we show that WFA is capable of binding to the S proteins of SARS-CoV-2, and therefore it potentially inhibits infection or spread of the disease. And can it be developed into an attractive therapeutic agent? Yes, I mean, either alone or in terms of prophylaxis in combination with drugs such as hydroxychloroquine, dexamethasone, or other treatments. And as I said, that one of the clinical trials is already in there, and we are with the finger crossed thinking that it can come as a good treatment. And thank you, everyone. And here I conclude. Just in case you encounter a fracture in these COVID times, reunion is there. Just in case you're suffering from osteoarthritis and a lot of pain, then Joint Fresh is there. And thank you so much for giving me this platform to talk.
Thank you so much, Dr. Renu Tripathi. In fact, I think all of you would agree with me that all the three lectures that we heard in the past 2 hours have been so enriching with information. I want to thank all the three speakers for giving us a treat of such a rich cerebral treat of information. It's a coincidence that the first lecture that started with qualities of ashwagandha, the following speakers, they vetted the claim and efficacy of ashwagandha emerging as the important botanical, the various have such a positive and promising role in COVID times. Thank you very much, all of you. And once again, I thank NASI headquarters and the local chapter Lucknow for giving us this opportunity of meeting one and all from NASI family at this platform. Thank you. Over to Dr. Suchitra Banerjee. Namaskar.
Thank you very much, Professor Veena Tandon, Madam. Now, after receiving the inspiring and innovative directives from Honorable Professor Padmanabhan Sir and Professor Manju Sharma'ji, followed by the informative lectures of all the three eminent speakers, it would be most desirable to reach at some conclusive follow-up plans to tackle this dreaded disease. Now, probably you all will agree that to fulfill this objective, there couldn't be a better person than Professor Paramjit Khurana to sum up the scientific session. So before I invite her to sum up the entire procedure, it is customary to introduce, though she doesn't need an introduction in the scientific fraternity. Professor Paramjit Khurana is an eminent plant scientist, plant biotechnologist, molecular biologist, and her research interest is to understand the processes of plant embryogenesis and heat stress tolerance in wheat, mulberry genomics, gene function identification, and allele mining for abiotic stress tolerance. She also played a key role in rice genome sequencing project and International Wheat Genome Sequencing Consortium. She has published more than 125 scientific papers in highly reputed journals like Nature, Science, Plant Biotechnology, Plant Molecular Biology, so on and so forth. She is recipient of Professor J.C. Bose Fellowship. She is fellow of all the three academies, including and additionally the World Academy of Science, Italy 2016. Professor Khurana is recipient of many, many awards. To give just a glimpse of them, I can mention few of them. Professor Archana Sharma Memorial Award from both ISCA as well as from NASI. Sri Ranjan Memorial Lecture Award from the National Academy of Sciences. Bharat Ratna Rajiv Gandhi Mahila Shakti National Award 2013, conferred by the Grassroots Studies and Research of India in collaboration with Rajiv Rural Development Foundation, Tirupati. Street Memorial Lecture Award by PCTOC. Certificate of Honor awarded by the Ganantri Sanstha of International Women's Day 2011, and there are many, many more. So with this brief introduction, I now request Professor Paramjit Khurana to just sum up the entire scientific session, please.
Thank you very much, Suchitra. not only for the introduction, but more so, I would say, for organizing such a spectacular seminar on such a topical subject. I think when I read the subject, bioresources for combating COVID-19, I was not very sure, but I see a complete amalgamation, not only of the audience, where we had luminaries, we had several past presidents, and who's who of not only the Lucknow chapter, but the national level also, and how you had organized the program. I think it was a very well articulated introduction also to the theme, wherein you had mentioned the role of NASI, especially Madam, how she had steered this program of the POSHAN Abhiyan via NASI through chapters and how everything was done. I think it was a wonderful organization, a good introduction of how the various chapters of NASI work under this year. The best thing which I liked about this particular program is, of course, the opening remarks by Madam Manju Sharma, who very rightfully said was responsible for bringing biotechnology to India. And I would say further also take it further that she is not only an institutional builder, but wherever we are today, I think India is the only country which has a separate scientific department of biotechnology. And she headed that particular department. I think hats off to her and a spirit for motivating all of us to get into the right path. And as mentioned by a president also, that it is not that she sticks with one topic. She keeps on moving with the changing conditions and always rises up to the challenges which are there. That is why I think she very rightfully mentioned that it is time to integrate biodiversity and bioresources and to shift from an industrial bioeconomy to a bio-industrial revolution. I think I regular stance there, and how science and technology should now be contributing to a bioeconomy of the country. I think that addition of that bio is very important here. So I think NASI is actually acting like a catalyst in bringing together these people of various disciplines and making them hold hands. You know, they could be health workers, they could be others, and so forth. That is where I think our president's remarks were very well timed. We couldn't have had a better president of the academy at this time. He, of course, if one is established, the opening remarks were given by one who established biotechnology in the country. I think our president is essentially responsible for all the R&D technology in the country itself. Was one of the first ones, and I think he gave a very holistic perspective of how COVID cannot be tackled by vaccine alone, and how development of a vaccine may not be the ultimate solution. And the ultimate solution may arise through decreasing of the inflammation and the cytokine storms via nutrition, which will enhance your immunity. I'm also particularly happy that he emphasized bioavailability and the role of bioavailability via nutrition and how that can. Because in many of our POSHAN programs and others at various other platforms, we have been talking, at least I have been emphasizing on bioavailability of nutrients, whether it is micronutrients or macronutrients as well. But then I think the best work was done by the Tandons. I would say the Tandons together, because both Professor Tandon, he not only welcomed us, he steered the grouping together of so many people here, coming here, and handling so many past presidents together and others, I think can be very challenging as well. But the best part was done by Sabine, who introduced all the three main speakers, and the topics of the presentation were in such good sync that she rightly mentioned that it indeed turned out to be a cerebral treat for us today. I was very enamored, I would say, by the presentation of Dr. Madhu Dikshit, who rightfully is the national chair at THSTI, and who very lucidly and very beautifully explained the role of drug repurposing and repurposing of a drug as common as ashwagandha. I say common because it is part of our ethnopharmacy and pharmacopoeia as well, and how it can be used for treating the new drug. To follow it up, I think Dr. Anil Tripathi added aroma to the subject. I would say, as to how bioresources can be used. He added aroma not only literally, figuratively also, but he also showed the mechanism of action as to how by the aroma action of the essential oils, the aerosol activity of the virus can be attacked. I think that was a very good point which he brought out, and how we could use for not only the intranasal cavity for nebulization and others, but even in the aerosol, how certain essential oils like garlic and others, which are known in our literature, but he showed the scientific evidence for that. And I think that was very good. He talked of other organic, I would say organics for combating COVID infection, like the Artemisia and of course Vibania and others, and also discussed the pros and cons of different formulations which are advocated by different countries, by different people, and what could be the possible repercussions. I think that a little bit of caution needs to be taken, especially when you're talking of karas and others of different things. I think that was very nicely brought out by Dr. Tripathi. So thank you very much for these presentations. And I think we started with Vibania, we ended with Vibania. I said we had an amalgamation of people here, but literally Ritu Trivedi talked of reunion, reunion of the bone, of the mineral. I think that's a topic which is very difficult because it involves a lot of musculoskeletal combinations, and how mineral density is important here, and how you can use plants for reunions as well. And compounds, products like Joint Fresh and others, which improve the proteoglycan layering of the joints also. I think this is the first time I've heard this presentation, although I'm aware of the work. I've seen many posters also at various fora, but it was a pleasure, I would say, overall to talk of it. And that last take-home lesson, which I think I should take it more personally, how she said how obesity will increase the sensitivity to COVID and other diseases. I think it's time for us all to take a serious scientific note of it. It is not that everything in abundance is good. Sometimes it may not be good as well. So ending on a lighter note, I think I would like to, that was the essence which I got of it, of how plants and medicinal plants can be repurposed and used for different purposes, and how we can actually, I think it was a very wonderful organization by Suchitra. Hats off to you for bringing in so many varied people from different disciplines, not only from Lucknow, but other areas. Also, I see people coming from Jaipur and other areas also. That is the good thing of the webinars, that you don't have to travel. The bad thing is that there will be many who are sitting on an empty stomach and wanting me to shut down. So I think I should do that well before your sugar levels go down. But I think I thoroughly enjoyed it. It was a wonderful treat. Hats off to you. And I look forward to more such presentations, not only by the Lucknow chapter, but by other chapters as well. So my personal thank you to the organizers, to the participants, to the presenters and others. Thank you very much. Over to you, Suchitra.
Thank you, Professor Paramjit, for extracting the whole fragments and the scientific relevance of organizing the webinar. It's an honor for us that you have summed up so well. Now, I would request Dr. Neeraj Kumar, Executive Secretary NASI, to extend the vote of thanks, who will give the vote of thanks to everybody. But I want to extend our vote of thanks on behalf of myself as well as from the NASI Lucknow chapter, who is the silent driving force to make this webinar happen. Thank you, Neeraj. Now over to you. Neeraj, please.
Thank you, Dr. Suchitra ji. First, hats off to you for organizing this very successful seminar. Actually, this seminar, the topic of itself, which was proposed by Madam Manju Sharma'ji, bioresources for combating COVID-19. She knows how to lead the team. She knows how to drive the forces into a particular cause, for the betterment of a particular cause. And I would like to say about Madam that since her joining this academy as an esteemed fellow in 1980, just after a few years, she started contributing to this academy with all means. And it is almost 35 years we are having her guidance as an institution in herself. She is an institution, no doubt, and icon for this academy, a torch bearer for this academy. And because all these programs which we are having in this academy are because of her incessant efforts and because of her valuable guidance which we are having regularly. Many thanks, madam, for your kind support, guidance, and all the energy which has been created by yourself in the academic arena. I am also very much grateful to our Honorable President, Professor G. Padmanabhanji. Actually, he is the person who is rightly dealing in this area and proposing many things, even the remedies, even about the vaccines. His first article, which came in March itself about this COVID-19, was enough to pave the way for how the research should go on in this country to give us the solution as early as possible. So, sir, your valuable guidance, your research, eminence, and everything is that is must be praised and must be given a good hand for applause.
Thank you.
We are very much grateful
Thank
to you, sir. This program, actually, this was held under the auspices of the Lucknow chapter and Professor Pramod Tandon, sir, who is the leader in that chapter and chairperson of that chapter. He works a lot for the dissemination of the scientific information and the know-how among the Lucknow and nearby areas with the support of secretaries like Dr. Suchitra ji and other advisors. Dr. Rudra Tripathi ji is also here, and they all support him. And with the support, naturally, the better half, Madam Veena Tandon ji, also this chapter is doing very well, and this is one of the example of the organization, a very good organization of the programs which we are already having. Sir, many thanks to you, sir. Professor Veena Tandon ji, she very in a very brief and interactive way, she managed the whole scientific session. I mean the different presentations, and not only gave the exact introduction in a very clear way about all the eminent speakers, but also gave hints about their speeches, what they are going to speak beforehand, just giving a guideline for the other participants, especially the students, how they are going to be benefited from the lectures of the eminent speakers. I am very much thankful to you, madam, on behalf of the academy and on my own. Thank you, madam. The eminent speakers like Professor Madhu Dixit ji, she gave a very nice informative lecture and conceptual lecture on the drug repurposing, especially the immunomodulation part of the ashwagandha. And it was a very rich presentation, full of the recent researches, how this ashwagandha, by the various people, how different sectors are taking this wonder drug for the COVID-19 and other diseases also. We are also very much grateful to Madam Madhu Dixit ji. Professor Anil Kumar Tripathi sir, his lecture on potential of essential oils and plant ingredients, especially on the eucalyptus, garlic oils, basil, cinnamon, artemisinin, all these plant essential oils which he emphasized in his lecture and their role about the modulation and the ingredient for the COVID-19 to fight against the COVID-19 was very clearly depicting the role of different essential oils in the different types of disease and the combination of those also for combating the COVID-19. We are very much grateful to you, sir. Thank you very much. Dr. Ritu Tripathi ji, her actually the subject is slightly different.